Hello everyone, my name is Shraddha and welcome to my YouTube channel Note of Biology. Today we will be discussing our last common structure found in all types of cells, the DNA or deoxyribonucleic acid. And we will discuss our first organelle in the series of cell known as the nucleus, which is of primal importance in case of eukaryotic cell. I have already discussed other common structures such as plasma membrane, cytoplasm, and ribosomes in my previous videos. In case if you have not seen those videos, please follow the link in the description box. Everybody recognizes this molecule, am I right? This is the DNA or deoxyribonucleic acid. By end of this video, I'm sure you will be able to identify the key structural details of the DNA molecule, where they reside in a prokaryotic or a eukaryotic cell, and what function the DNA plays in a cell. DNA or deoxyribonucleic acid is the basic unit of heredity. What makes us tall, what gives color to our hair, all is coded in the DNA. It is also known as the information center of a cell because it stores the instruction for making molecules such as proteins. In case of prokaryotic cells, because they don't have a nucleus, the DNA is found in the cytoplasm in a region known as nucleoid. In eukaryotic cell, the DNA is separately located in a membrane-bound organelle known as the nucleus. Now, the first person to isolate the DNA molecule was the scientist Friedrich Miescher. In 1869, while studying the WBCs found abundantly in pus, he observed that the nuclei of WBC contained a substance rich in both phosphorus and nitrogen, and thus the first DNA molecule was known. Later, other scientists established the fact that DNA is responsible for transmission of hereditary information from the parent to the offspring, and that it is a polynucleotide. Now, in order to understand the biological function of the DNA molecule, we need to first understand its composition. In the previous slide, I said that DNA is a polynucleotide, which means it is made up of multiple mononucleotides. In simple words, just like a polymer which is made up of multiple repeating units of monomers, a DNA is a polynucleotide made up of repeating units of monomers known as nucleotide. Now, each nucleotide is made up of three key things: a sugar, a nitrogenous base, and a phosphate group. In case of the DNA molecule, the sugar is a 5-carbon sugar or the pentose sugar known as deoxyribose. The nitrogenous base can be divided into two categories, purines and pyrimidines. The purines are larger than the pyrimidines because purines have two ring structure and pyrimidines have only one ring structure. The examples of purines include adenine and guanine, and the examples of pyrimidines include cytosine and thymine. The nitrogenous bases are always attached at 1' carbon of the sugar. The phosphate group are always found in between 5' carbon of one sugar molecule and 3' carbon of the neighboring sugar molecule. DNA exists in the form of a double helix where the two strands of the DNA are intertwined into each other and that they are connected via bridges, just like a spiral staircase where the two sides are intertwined into each other and in between they are connected with the help of stairs. Each nucleotide, as said, is made up of a sugar, phosphate, and nitrogenous base, and these nucleotides that make up the DNA are attached to each other with the help of phosphodiester bond. The sugar in the DNA molecule is known as deoxyribose because it has a missing hydroxyl group at the 2' carbon of the sugar molecule, and thus the nucleotides in the DNA molecule are known as deoxynucleotide. The double helical model of DNA molecule was proposed by Watson and Crick in 1953. They stated that the DNA molecule is composed of two anti-parallel polynucleotide DNA strands that form a double helical structure. The backbone of the DNA molecule is made up of sugar and phosphate, and this sugar and phosphate make up the outside of the helix, while the nitrogenous base forms the inside of the helix. The hydrogen bonding between the nitrogenous bases on the opposite strands is responsible for holding the two strands of the DNA together. In each helical turn, which is around 3 to 4 nanometer in length, there are approximately 10 nucleotide base pairs. Nitrogenous bases that form base pairing are very unique. They are not just random. The adenine nitrogenous base always pairs with the thymine, and similarly, the guanine always pairs with the cytosine. These ATGC associations are known as complementary base pairing. After discussing the hereditary unit of the cell, the DNA, now let's move forward and learn how the eukaryotic cell stores this important piece of information into a membrane-bound organelle known as the nucleus. A nucleus can be defined as a membrane-bound organelle in an eukaryotic cell which contains the genetic information of the cell, the DNA. In case of a eukaryotic cell, the DNA never leaves the nucleus, rather it is transcribed into RNA molecules which then leaves the nucleus and travels into the cytoplasm. Inside the cytoplasm, upon arrival, the RNA molecule helps the two ribosomal subunits, small and large, to combine together and undergo a process known as protein synthesis. If you haven't watched my video on protein synthesis and ribosomal unit assembly, please follow the link in the description box. Now let's discuss the structure of the nucleus in detail. The nucleus is completely bounded by membrane and it is the characteristic feature of a eukaryotic cell. However, there are certain exceptions like osteoclasts, which have multiple nuclei, or RBCs, which even lose their nuclei upon maturation. Just like every organelle which is bounded by membrane, the nucleus is also enveloped with a membrane known as the nuclear membrane or the nuclear envelope. This envelope is made up of phospholipid bilayer, just like we had in the plasma membrane. It has an outer membrane and an inner membrane, and this nuclear membrane helps distinguish the contents of the cytoplasm from the nucleus. Inside the nucleus is found a gel-like matrix which is known as the nucleoplasm, which contains the DNA either in the form of chromosomes or chromatin. Now chromosome are the dense structures which are seen when a cell is ready to undergo cell division. However, when the cell is functioning normally or is not ready to divide, the DNA exists in loose thread-like forms known as the chromatin. In the image shown on the right side of the slide, we see the blue colored threads which are the chromatin. Now inside the nuclear membrane there are certain pores which act as a trading port and allow certain substances to move into or outside the nucleus. This is known as nuclear pore. Another key structure which resides in the nucleus is known as the nucleolus, which is responsible for the formation of ribosomal subunits in a eukaryotic cell. The location of DNA in a prokaryotic cell is very different from a eukaryotic cell. Since a prokaryotic cell lacks nucleus, the DNA is located in the cytoplasm of the cell in a region known as nucleoid, and the DNA is arranged in the form of circular chromosomes. In case of eukaryotes, as we learned, the DNA is housed in a membrane-bound organelle, the nucleus, and therefore it exists inside the nucleus in the form of linear structures. The functions of the nucleus in a eukaryotic cell includes: it contains the hereditary information of the cell, that is the DNA. It acts as a site of synthesis of ribosomal subunits. It also coordinates and controls many important cellular processes of the cell, such as cell division, protein synthesis, and so forth. A 1-minute summary of DNA and nucleus would include the following points. DNA is a polymer made up of nucleotides. Each nucleotide is made up of a pentose sugar, a nitrogenous base, that is adenine, guanine, thymine, or cytosine, and a phosphate group. The nucleotides are held together with the help of phosphodiester bond. The double helical model of the DNA was proposed by Watson and Crick, who stated that DNA exhibits complementary base pairing, that is, A pairs with T and G pairs with C, and that the two strands of the DNA molecule are held together by a hydrogen bonding. In case of a prokaryotic cell, the DNA resides in the cytoplasm of the cell in a region known as nucleoid. However, in case of eukaryotic cell, it is located in a membrane-bound organelle known as the nucleus. The function of a nucleus in the case of eukaryotic cell is primarily to store the hereditary information, the DNA. For future videos, please like, share, and subscribe to my channel, Node of Biology.